Okay, so island systems are well studied and they serve as great models to understand diversification. And one of the main reasons is that on islands, the number of factors promoting divergence is usually less than that on continental systems. So we can sort of narrow down the number of factors promoting divergence, simplify and understand a little bit better. And one island that has been relatively well studied is that of Sao Tome in the Gulf of Guinea. So Sao Tome is a small island off the west coast of Africa, kind of in the armpit of Africa there, and it's part of the Cameroonian volcanic belt. And I like to think of the island of Sao Tome as a pulperia. It's rather small, but it has a lot to offer. You can giggle, it's all right. So Sao Tome is a little bit smaller than the state of Rhode Island, but the reason it has a lot to offer is it's created a lot of different interesting patterns of diversification. The reason is that it's rather heterogeneous across the island. So small, but a lot of heterogeneity across the island. So one example of that heterogeneity is this gradient of precipitation. So we see that we have a lot of precipitation on the southeast part of the island, over 3,500 millimeters, and then 1,100 millimeters per year of precipitation on the other side of the island. And so we have this rather extreme gradient of precipitation across. And the reason is because you have this large volcano in the middle of the island that creates a rain shadow that kind of blocks the rain from moving across. So this could promote divergence in taxa along some sort of a gradient. Alternatively, we could think about how the geology affects divergence across the island. So there's two main rock ages on Sao Tome. We have younger rock that's about 1 million years in the north, and then older rock 2 to 8 million years in the south, with this really discrete break in the middle of the island. And so this different rock ages could have promoted different substrate types, and taxa that are really sensitive to differences in substrate could be affected in their divergence history due to this geological history. And then I'll mention that this dark blue section is uncharacterized. We know very little about this corner of the island. So I won't have samples from there. Other studies don't really have samples, and it's fairly unknown still. And then finally, we can think about really contemporary processes like land cover, and particularly how humans have influenced evolutionary history on the island. So here we've categorized land cover into 4 different colors. So we have yellow is human settlements, green, generally agriculture, the kind of intermediate lighter blue is secondary forest or transitionary forest, and then we have that dark purple is the primary forest. So we have kind of this humanized, modified half of the island, and then the other half is relatively forested and undisturbed. And this could have had a lot of impacts on secondary contact or hybridization and some taxa. All right, so something else that's really exciting about Sao Tome is that there's a high number of endemics. So it's a remote oceanic island, never been connected to the mainland, fairly far from the mainland. So this is expected that a high proportion of taxa would be endemics. And so we have begonias, the flowers in the top, lots of begonias, some of the highest fern diversity in all of Africa, 4 genera of birds, a lot of bats. But what's sort of unexpected about the endemism of Sao Tome is that we have this shrew species, a lot of frogs that are endemic, a bunch of fossorial and small reptiles, and then this amphibian that we're talking about today, the caecilian, the Sao Tome caecilian. And these are all species that are very dispersal limited. Oftentimes they spend most of their lives underground, not things you would expect to be arriving at a small remote island off the coast of Africa. One other pattern that's really interesting on Sao Tome is despite its small size, probably because of that heterogeneity across the landscape, there are a couple documented cases of hybridization. So one really well-studied case is that of Drosophila, and then there's also one example of a frog species, or frog species group, Hyperoleus. So you have a lowland species that specializes in open habitat, well, not entirely lowland, but generally open habitat, more generalist species. And then a species that's more of a forest obligate that depends on forest cover. And then where they intersect or come into contact, we observe hybridization. So the species I'm focusing on today is Schistostomotopum tomensi, the Sao Tome caecilian. And I have been practicing sounding that out because it's rather difficult to pronounce. But this caecilian is sort of like the model caecilian in a way because we know almost nothing about most caecilians. But this caecilian is pretty well studied. So caecilians are an order of amphibians, but they're mostly fossorial, mostly in the tropics, and so they're just not well understood. But this species is pretty common on the island of Sao Tome. So those who have been there find them very commonly. We have a lot of samples of them, and there's been a number of studies. But what makes them interesting to study diversification is that they're fossorial. And so we don't know a lot about fossorial species and their diversification history on islands. So a little of the taxonomic history of this species in order for us to set up our hypotheses. This species was described in the late 1800s, and it remained as one species on the island until 1965, when a researcher, Taylor, was looking at museum samples, and he noticed that some of the specimens from the south had this black speckling. And so based on this color morphology, or difference in color, and then also some head shape differences, he described this southern group, the speckled individuals, as a separate species, Elaphey. But then Nussbaum and Frender, it remained that way for a few decades. And then they, in the 1998, they visited the island. They surveyed across, particularly the eastern coast, and when they laid them out in sausage form, they found that, or they hypothesized that this wasn't in fact two different species, but rather this sort of morphological gradient, or a gradient of morphology. So they found that in the middle of the island, you had individuals that had intermediate levels of speckling, which they hypothesized was a gradient from northern to southern individuals. And so they kind of laid out two hypotheses. In the first case, we have one species with this morphological gradient, and the reason we see intermediate individuals is it's just varying with this heterogeneous landscape across the island. And then the second hypothesis would be that we have two species that are intersecting at some point in the middle of the island and potentially hybridizing. And the reason we have intermediate morphologies is that these are actually hybrid individuals. And so these hypotheses were tested initially using genetic data by Stolting et al. In 2014. And they generated mitochondrial data. And what they found is that they had this northern haplotype group, which is in red, and then a couple of southern haplotypes, but generally one southern group with the other three colors. And so they found evidence of two lineages on either side of the island that come into contact in the middle and are putatively hybridizing there. So that's right here. We have all of these localities that have both haplotype groups in the middle of the island. And they pointed out that this correlates really nicely with this geological layer or substrate types, where you have the intersection of these two ages of rock. And so they hypothesized that this geological history or differences in substrate may have promoted divergence and potentially hybridization in this group. But in the absence of nuclear data, they were really unable to test for the presence of hybrids. And so they supported this hypothesis that there's two species potentially hybridizing in the center of the island. And so our group came along and we added a little bit of additional mitochondrial data for a few localities that have been collected more recently. We scored all of our individuals that we have genetic data for morphologically to check the morphological scoring of past studies. And then I generated some RAD data I'll show you data for 43 individuals today. So starting off with the mitochondria, we've already seen this haplotype network, but we have evidence of a northern and southern lineage, possibly admixing in the middle of the island, and then these are the localities we added, so there's been some additional fieldwork done in the south, and then one additional site in that possible admixture zone. When we look at the morphological data, we see that we have unspeckled individuals, and those would be the purple circles, mostly in the north, and then mostly speckled individuals in the south. And because we were using museum samples, we were unable to score individuals as intermediate levels of speckling, and so we just scored them as speckled or unspeckled. We can see that for the most part, the two morphotypes come into contact at that same area in the middle of the island where we had possible mitochondrial possible admixture. Not perfect. There's a few individuals that are kind of outliers, but mostly they come into contact in the middle. And then when we look at the RAD data, I used admixture to estimate population structure. And then I sorted my admixture plot from north to south so that we could see where admixed individuals are. And we see that the nuclear data supports the other two data types with this possible admixture zone pretty much in the same spot in the middle of the island. So when we put the three data types together, I put this orange box around individuals that are in the geographic locality of that possible admixture zone. So we have individuals that are pure northern and pure southern on the other sides, and then see that all of the possibly admixed individuals are in that zone, that contact zone. If we layer the mitochondrial data on top, we see that there's a bunch of examples of mitonuclear discordance, but all of them are within that possible admixture zone. And we look at the morphological data, we see that the contact between the two morphotypes is also, as we saw earlier, within that area. So we see evidence for hybridization within this area across 3 different data types. And so all of this data together supports the hypothesis that we have a northern and southern lineage that are hybridizing in the middle of the island, although we're kind of following up the hybridization hypothesis with some more quantitative tests. So now we can go a little bit further. We've looked at the patterns of diversification, but we can think about what processes promoted these patterns. And so we can ask how and when did divergence occur on Sao Tome? And we can bring it back to these layers that we hypothesized could be promoting evolutionary history or affecting divergence on the island. Specifically, if we would expect to see a gradient across the island, then something like precipitation could be promoting divergence. And then the age of diversification, if there are 2 lineages, would support one of these other layers. And so I used Dottie to conduct demographic modeling. And I tested a couple of simple hypotheses that varied in the level and timing of gene flow. So the first model was just divergence with no gene flow, then divergence with continuous gene flow, divergence with gene flow followed by a period of isolation, and then divergence in allopatry with secondary contact. And the best supported model was divergence in allopatry with secondary contact, and the parameters Dottie gave suggest that the secondary contact was in the recent past, and it was a strongly supported model. So then I used GFOX to try to estimate when this divergence in secondary contact occurred, and GFOX suggested that divergence occurred about 250,000 years ago, with secondary contact very recently at 24,000 years. And so this rules out a Klein hypothesis because we have evidence for 2 lineages, so we can rule out precipitation. Humans have only been on the island affecting a land cover for about 5,000 years, so this rules out land cover, and it leaves us with, at least for now, a hypothesis of the geology or the rock age and the substrate promoting divergence and secondary contact in this species. And particularly, maybe the volcanic history in the recent past has also affected some of these patterns. So with that, I'd like to acknowledge the Bell Lab, Matt Fujita's lab for sending us some reagents, and all of the support staff at the National Museum of Natural History for all of their help. And I'll take any questions.
What's the nearest relative on the mainland?
The nearest relative on the mainland is there's 2 species in the genus, and the sister species is in East Africa. So in many cases on Sao Tome, the sister species are in East Africa. They're thought to have rafted on vegetation rafts to Sao Tome. What else? Yeah, right here.
So can there be expansion before secondary contact? So instead of just geology, it can just be population expansion.
Expansion.
Parental expansion before secondary contact.
Okay. Yeah, I don't know. I haven't tested for size change.